Okay. Sit straight, listen all. Biomolecules we are going to start up. This is not a small chapter, not a big chapter, but a moderate way you have to deal with it. The content of this chapter is moderate. Okay. So, wherein from the beginning of this chapter, if you have to see biomolecules, what are we learning in this chapter? At the beginning, you will learn on elemental analysis. That means comparing the elements of a human body in a living organism and the elements that are present on the earth crust. And later you will learn on chemical analysis. When you are learning chemical analysis, you will learn on what is acid soluble pool, what is acid insoluble pool, what are the molecules that are landed to acid soluble pool, and what are the molecules that are landed to filtrate, like that, retentate and filtrate. This is what you will be learning in chemical analysis. After that, you will be learning on ash analysis. What is ash analysis also we will discuss. These are very minute introductory segments of this chapter, but we have to address it, because if statement based question comes from the introduction, then we should not be. So, this is about elemental, chemical and ash analysis. So, after that we will be learning on primary and secondary metabolites. Primary and secondary metabolites, very important. Secondary metabolites, one table is given in your NCERT, right? Some 3 to 4 times the questions are asked on that table, only on that table, okay? And mostly from biomolecules, you will get questions, like if we have to take 10 PYQs, 5 to 6 will be from tables of biomolecules, okay? So, that is about primary and secondary metabolites, and then you will start learning on biomacromolecules, okay, which includes carbohydrates, proteins, nucleic acids and lipids, okay. So, this is the first half of the picture, biomolecules. If anybody want that framework, please take it, whatever I am writing here, you can take that framework. So, whenever you are going with the revision of this chapter, you can tick it and move forward with this chapter, okay. So, what is that? First we are learning elemental analysis, and this table is very important. Second will be chemical analysis, its procedure, and what are you going to get in acid soluble and acid insoluble pool, that you have to identify. And third is ash analysis, you will be learning, and then differences between primary versus secondary metabolites. Right, and then you will start on learning with the biomacromolecules, which are obtained in acid insoluble pool. I will explain you it clearly, but as of now remember, this is the beginning of this chapter beta, and here in acid insoluble pool, what are you getting? Like carbohydrates, all the biomacromolecules, proteins, lipids and nucleic acids. So, here from carbohydrates, what you will be learning? Types of carbohydrates, okay. Carbohydrates usually are addressed as saccharides. Based on the number of carbon units, you have monosaccharides, disaccharides, trisaccharides or polysaccharides, right. So, all the different types of saccharides, the bonds that are involved in between two sugars, what are reducing sugars, what are non-reducing sugars, that and all you will learn where under carbohydrates. Proteins, the interesting part. Amino acids are building blocks of proteins, we know, and all the amino acids are different. There is nowhere you can see homopolymer of protein chain. Polypeptide chain is never a homopolymer. It is a heteropolymer chain, because we have different types of amino acids, right ma? So here, when it comes to proteins, we are also learning on different structures of proteins. Okay, our body is built up of many protein structures, and we intake proteins more than carbs these days. We prefer more intake of proteins than carbs. In what structures you can see these proteins as? Primary structures, secondary, tertiary, quaternary structure. Example of quaternary structure here, hemoglobin with 2 alpha and 2 beta subunits. Right, Ra? Understood? So that's what we are going to learn under what? Proteins. And there is a big table given on proteins also. We can see transporters, we can see channels, we can see hormones, everything is a protein, right? And then lipids. Lipids also we are going to learn different types of lipids. 4 types of lipids we are going to learn. And then nucleic acids. The question on nucleic acids can be combined with your molecular basis of inheritance, because a nucleic acid is a polypeptide chain. Polynucleotide chain. Each nucleotide will have 3 components. What are they? Sugar, phosphate group and nitrogen base. You have different types of nitrogen bases again for RNA chain and for DNA chain. So that and all you have to learn. Okay, if you are thorough here, there it will be easy, right? So this is the first half, only first half of the picture. Second half will be completely on enzymes. If you take the previous year questions, 40% of questions will be on enzymes from this chapter. 40% of questions will be from enzymes. From enzymes, what you are going to learn? First, its nomenclature, not completely, but how the 4 digits are framed. Till that if you know, it is enough. Nomenclature. Second is classification. We have 6 classes of enzymes. We need to learn with examples. Okay. And third is mechanism of action. We have 2 mechanisms of enzymatic action. One is lock and key hypothesis. Second is induced fit model. Lock and key is static model. Induced fit model, like how your hand will be going into gloves, same the way. Okay, so those 2 mechanisms you have to be clear. Mechanism of enzyme action. After nomenclature, classification, mechanism of enzyme action, you have to know what is transition energy and that chart which is given with enzyme substrate complex in the presence of enzyme and in the absence of enzyme. That graph is very important. After which at last you will be learning on inhibitors, and last segment is cofactors, the non-protein part of the enzyme, right? Protein part is known as apoenzyme, non-protein part is cofactor, which is of 3 types again. It can be metal ion, it can be coenzyme or prosthetic group, right? So this is the complete framework of the chapter. Got an idea of what we are learning this? Might be you already know few of the topics, but it should be in a proper structure so that you can revise it properly. So once in a gist, in a minute, what are we learning in this chapter is starting with elemental analysis, chemical analysis, ash analysis, and then from chemical analysis, whichever biomacromolecules we are getting into, that we are learning as carbohydrates, proteins, lipids, nucleic acids in detail, after which we are learning on enzymes, right? Its nomenclature, classification, mechanism of enzyme action and inhibitors, lastly cofactors, right beta? Clear till here, right? And we are going to start with elemental analysis first. Very simple on this. When the elemental analysis have performed in a living organism and taken a small amount of earth crust, all the elements are same. Whatever are present in the earth crust are also present in the living organism, but their composition is different. When it is observed in a closer way, the composition is different. What are abundant in living organisms? These are the liners of your NCERT. All the elements present in the sample of earth crust are also present in the sample of living tissue. All the elements are there, but the relative abundance of carbon and hydrogen with respect to other elements is higher in living organism when compared to earth crust. Very important statement. Which elements are abundant in living organism? Carbon and hydrogen. But when you look into the table that is given, you can see few more elements which are abundant. You need not to remember the exact figures, but you have to know which is abundant either in earth crust or in the living organism, right? So here we go. First we will see about hydrogen. It is abundant in human body. It is 0.1 here, it is 0.5 here. 0.14 in earth crust and 0.5 in percentage in human body. And then second carbon, if you see, it is 0.03 in the earth crust, 18.5 in the human body. So it is more in the human body. Then oxygen, if we observe, 65.0, here 46.6. So relative abundance is more in human body. Then nitrogen, also abundant in human body. Sulfur, abundant in human body. Sodium in earth crust. Calcium in earth crust. Phosphorus, human body. Magnesium, earth crust. Silicon, also earth crust, right? So this relative abundance, you just keep it in your mind. If you are noting it down anywhere with pencil also in your NCERT, you can. Okay, when you are revising, what are abundant in human body? If you remember enough, the rest will always be an option, right? So this is about elemental analysis. Clear, Noma? Simple and easy. Yeah. Next is chemical analysis. Yeah. So here in chemical analysis, a living tissue, any living tissue, you can take a living tissue of an animal or a plant, so any mesophyll tissue, any leaf you take, and then you have to grind it with mortar and pestle. Anytime in your kitchen if you have seen, but different kind of mortar and pestle we have to use in lab. Mortar and pestle, what chemical we are using? Trichloroacetic acid. Then all the components that are present in the tissue will be entered into what? Solvent, that is trichloroacetic acid. Then you will be taking it into a test tube, right? You will not dump it as it is into the test tube, you will filter it with a cheesecloth. The thick slurry is obtained and you have to strain it with a cheesecloth. Then inside the test tube, you can see after centrifugation, means that will be centrifugated like 3000 rounds per second or 5000 rounds per second, and after that you can see at the base, that is retentate. This is what ma? Retentate. Retentate is also noticed as acid insoluble pool. And the upper part where only lighter molecules will be fall into, this is addressed as what? Filtrate, which is known as acid soluble pool. Most of the cellular constituents, inorganic molecules, organic compounds will be landed into filtrate, okay, whose molecular weight is less than 800 Daltons. Okay. So what you will observe in the acid soluble pool? Acid soluble pool you can come across most of the inorganic molecules, cellular constituents, organic compounds, in simple terms. After this we will be learning primary and secondary metabolites. No, secondary metabolites also you will be seeing it in acid soluble pool itself. This differentiation or clarity is very important. Then what do you see in acid insoluble pool? And all this molecular weight, in how many Daltons? What is the molecular weight of this? It will be less than 800 Daltons of molecular weight, all the components inside a cell, right ma? In acid insoluble pool, what are the molecules that are being obtained here? Carbohydrates, proteins, nucleic acids, and lipids are also visible under acid insoluble pool. All these are known as what ma? Biomacromolecules. Right? All the biomacromolecules are obtained in acid insoluble pool. Biomacromolecules or biomolecules one and the same. How they are different from this? Can anybody tell me? First is molecular weight. It is around 800 to 1000 Daltons. 800 to 1000 Daltons or more than 1000 Daltons. More than 1000 Daltons. Above 800 or more than 1000 Daltons are in here. But there is one more difference. All the carbon containing compounds are known as biomolecules. All the carbon containing compounds inside the living organism are known as what? Biomolecules. In biomolecules we have macromolecules, micromolecules. Macromolecules are those biomolecules which are obtained in what? Acid insoluble pool and whose molecular weight is above 800 Daltons. One more question, if you remember, okay, because I think this is second time you are going through this chapter. So, amongst this four which I have given here, one molecule is known as pseudo biomacromolecule. It is a not true biomolecule. What is that? Lipids. Why? Why? Why it is not a true biomacromolecule? Be clear. Telugu, Hindi, English, anything. Sab kuch chalta. Very good. Superb. It has less molecular weight when compared to other macromolecules. Then why do you see it here? Think. No, what she said is exactly correct. Its molecular weight is less than 800 Daltons. That is the reason it is not known as true biomacromolecule. That explanation is 100% okay. Then why I am seeing this here in the written date is my question. Second point is also coming from this row. Yeah? It is insoluble in water. Also fine. Why? See, when you are, we have learnt fluid mosaic model, right, in cell the unit of life. How much percentage of plasma membrane is composed of lipid? Let us say RBC, erythrocyte cell membrane. How much percent of it is? 40% or 42%, right, is composed of lipids. When it is grinded with mortar and pestle, they are like this, right, in lipid bilayer they are like this. But then when they are disrupted and grinded up, they will turn up forming into micelles. I think this you will also learn in your chemistry. They will try forming out what? Micelles like this. They will encircle it. And these globules are insoluble in water. Insoluble in solvents, they will just form up into globules and they will land up in what? Retentate. That is the reason you will see it in the retentate. The two explanations are also correct. Its molecular weight is less than 800 Daltons. It is insoluble in water. Why is it insoluble in water? Because when the lipid bilayer is grinded, it is disrupted. It is turning it into micelles and it is getting insoluble. Right? So, this explanation should be clear in your mind so that we are free in answering any type of question. Right? So, this is about chemical analysis. So, in chemical analysis, we will be using many solvents actually in reality, but let us go with the one solvent which is given in our NCERT. What is the chemical we are using? Trichloroacetic acid. What is the procedure? We will take a living tissue, grind it with trichloroacetic acid, and then we will take it into a test tube by filtering it with water cheesecloth, and the liquid which you are getting it, you will centrifugate it. After centrifuging, you will get two layers. This lower layer you will call it as what? Acid insoluble pool. Upper light molecules, these are known as what? Acid soluble pool. This is retentate and this is filtrate. What are the molecules that you can see in the filtrate? You can see inorganic molecules, cellular constituents, organic compounds and secondary metabolites whose molecular weight is less than 800 Daltons. And here if you observe, in acid insoluble pool or retentate, you can see biomacromolecules like carbohydrates, proteins, nucleic acids, lipids. These are the biomolecules inside a living organism, right? Those which we are consuming daily, right? And their molecular weight is more than 800 Daltons or more than 1000 Daltons. Any of the statement is correct only. In this, lipid is known as false biomacromolecule. It's not a true biomacromolecule because its molecular weight is less than 800 Daltons. It is water insoluble because it is turning up forming micelles. Remembered? Clear, right? Where do you see secondary metabolites? Acid soluble pool. Where do you see primary metabolites? What are primary metabolites? Okay, we are going to learn now. Till here to you are clear, right? Secondary metabolites you will see in acid soluble pool, inorganic constituents, cellular constituents, everything you will see here. Once look into theory also beta. Retentate contains biomacromolecules, molecular compounds like polysaccharides, nucleic acids, proteins and lipids. Filtrate or acid soluble pool contains biomicromolecules, means all the secondary metabolites, cellular constituents, inorganic molecules, organic compounds are known as what? Micromolecules, whose molecular weight is less than 1000 Daltons, or you can say in specific 18 to 800 Daltons. It is 18 to 800 Daltons. Usually entire cytoplasmic composition except biomacromolecules will be landed into the filtrate. Okay, clear now? Next. So, now we will see what is ash analysis and then we will move on learning differences between primary and secondary metabolites. Till here to everything is clear, Nama. Ash analysis is simple. Whatever living tissue you are taking, you will weigh it. That is known as fresh weight. If you are drying it, for example, I have taken a leaf extract. Okay, as I am used to it, I am always saying leaf extract. I have collected some leaves, have dried it under fan for 2 or 3 days or for a week. Okay, or else I have used some dryer and have dried all the leaves. Then I am weighing that leaf. Then what is the difference from fresh weight to dry weight? Evaporation. Water molecules will be lost and the leaf is completely dried. It is very much highly concentrated with the secondary metabolites or the constituents it has, because all the water molecule is evaporated. That weight is known as what? Dry weight. From that dry weight, if you are burning the tissues and all, what you will get? Ash. And that ash is analyzed, like how we are performing chemical analysis from a fresh weight of living tissue. Ash analysis is performed from a burnt living tissue. Okay, and so a small amount of living tissue is weighed, the tissue is dried, that is known as what? Dry weight, and the dried tissue is fully burnt. all the carbon compounds are oxidized. Okay, so in what forms? Carbon dioxide and water vapor, they are removed. What remains is called as ash. Whatever is remained in that burnt part is called as what? Ash. That ash is analyzed, and in that ash analysis you can see all the inorganic elements. What you can see? Inorganic elements. That inorganic elements include sodium, potassium, calcium, magnesium, water as water vapor or like water molecules, and compounds like sodium chloride, calcium carbonate, phosphate and sulfates. Right. So, this is the definition of biomolecules we have learnt, and this is what you will see in ash analysis. All these are what elements ma? Inorganic elements. Inorganic elements. In any paper of your previous NEET, there is never a question asked on ash analysis, but you should know, you should have an idea, that is all, right. So, you are clear on elemental analysis. Which elements are relatively abundant in human body when compared to earth crust? Which elements? Carbon and hydrogen. And in table if we see oxygen, few more elements if you can name. Nitrogen, sulfur. Okay. And in chemical analysis, what is the chemical we are using? Trichloroacetic acid, right. And what are the two fractions we are getting? First fraction is filtrate, also known as acid soluble pool. Second fraction is retentate, also known as acid insoluble pool. In filtrate, what you will get? What are the molecules you are getting? Cytoplasmic constituents, secondary metabolites, organic compounds, inorganic elements. And in acid insoluble pool or retentate, what are the molecules you are getting? Carbohydrates, proteins, lipids and nucleic acids, right. And what will be the molecular weight of acid soluble pool? Less than 800 Daltons. In acid insoluble pool, more than 1000 Daltons. Lipids, whose molecular weight is less, so it is not a true biomacromolecule, and it forms up micelles, it is insoluble in water, right. And then ash analysis. Ash analysis is performed from fresh weight or dry weight of living tissue? Dry weight of living tissue. And what are lost from that ash analysis? Carbon dioxide and water. The remaining inorganic elements you will get from ash analysis is this, right. So, this is about till here, and then we are learning on carbohydrates first. First primary and secondary metabolites. See, first of all, what is the meaning of the term metabolite? You think for a while, I will think what. What is a metabolite? You just think. What is a metabolite? You have seen plant physiology, you have seen glycolysis, TCA cycle, you have seen C3, C4 cycles and all. So, after every step you are getting an intermediate organic compound and some end products also you are getting. So, the intermediate compounds might be, end products might be, all that including the starter, they are known as what? Metabolites. Okay. All the molecules which are into the biosynthetic pathways are known as what? Metabolites. It can be primary metabolites or secondary metabolites. You see, metabolites are intermediates or end products of cellular metabolism or any biosynthetic pathway. Let us say glycolysis only. Glucose is a metabolite, it is a primary metabolite. Glucose 6-phosphate is a metabolite. Pyruvate is a metabolite. Fructose 1,6-bisphosphate is a metabolite, like that. Understood now what is a metabolite? Next, organic compounds including amino acids, sugars are called as biomolecules. They are known as what? Metabolites. They are of two types. One is primary and second is secondary metabolites. So, the initial difference is this: primary metabolites are those which will help in the growth and development of the living organisms. For example, the food you are taking, which consists of carbs, proteins, fats, which help in growth and development of living organism is what? Primary metabolites. Secondary metabolites are those which help in defense action. For example, all the phytolexins which are present in the plant, pigments that are present, all that are what? Secondary metabolites. Okay. So, here you see primary metabolites are chemical compounds produced during growth and development processes. Secondary metabolites are those analysis of plant, fungal or microbial cells. Several compounds are seen besides primary metabolites for different functions, be it for pigmentation, for defense mostly, those are known as what? Secondary metabolites. You got the initial difference, right? Now, these are involved in primary metabolic processes like what? Respiration and photosynthesis. Just now we have taken one example of glycolysis pathway. Secondary metabolites are not required for primary metabolic processes. Examples include what, ma? Lauder, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, all these are secondary metabolites. Primary metabolites includes carbohydrates, proteins, lipids, nucleic acids, right? So, this examples and definition, if you remember, it is enough. Okay, and the table that is given, this one. If you are pretty good at memory, you don't need any mnemonic, but for people like me, I need some mnemonics. This is one of the funny mnemonic that I can give. Pretty angry tigers eat tasty laddus during picnics. Like P for what? Pigments. Example, carotenoids, anthocyanins, xanthophylls, okay, chlorophyll, all these are pigments under P. Next A, alkaloids include morphine and codeine from your biology in human welfare. Next, terpenoids include monoterpenes, diterpenes, sesquiterpenes, all these are different types of terpenes. Next, essential oils, lemongrass oils, toxins, abrin and ricin, lectins, concanvalin A, might be some 2 to 3 times asked question. Drugs, vinblastine and vincristine, curcumin, you can take one more example of vincristine. Next, polymeric substances like rubber, gums and cellulose. Just if you remember this mnemonic, under P to pigments, alkaloids, terpenoids, essential oils, toxins, lectins, drugs and polymeric substances, right? Just two examples under each. Fix it in your mind now itself, okay? You need not to by heart and say to someone else. If you are in a position to identify, that's enough, right? Next, so all these are obtained where? In acid soluble pool. So now my question, I am getting back to my question. You have learnt what are primary metabolites and what are secondary metabolites. You have learned the examples also. And I told you secondary metabolites, you can see it in acid soluble pool. Where do you see the primary metabolites? Acid insoluble pool. Because all the primary metabolites are what? Like carbohydrates, proteins, lipids and nucleic acids, which are starting the metabolic processes like photosynthesis and respiration, right? So, just remember this. I hope you will not forget. This is about primary and secondary metabolites. Shall we get started with biomacromolecules? First, carbohydrates, ma'am. Yeah. So, this we have already learnt. Carbohydrates, you see. Carbohydrates are composed of what? Carbon, hydrogen, oxygen, that's all. Carbon, hydrogen and oxygen. CN, H2N, ON is the formula. CN, H2N, ON. For example, C6, H12, O6, glucose. This is the general formula for all the carbohydrates. It is composed of carbohydrates, hydrogen and oxygen. These pigments are also known as what? Saccharides, because their basic components are what? Sugar. They are of two types based on number of carbon compounds they consist of. It can be of Small carbohydrates or large polysaccharides? Small carbohydrates are again of three types. First is what ma? Monosaccharides. Second, derived monosaccharides, oligosaccharides, polysaccharides. Right? So first monosaccharides you see. These are the simple sugars which consists of how many carbons? 3 to 7 carbon atoms, that's all, which cannot be further reduced. 3 to 5 carbon atoms it has. Examples, you can take any 5 carbon compound, 6 carbon compound like glucose, fructose, mannose, galactose and hexoses. Here there is some description given which you have to remember. Glucose is also known as what? Dextrose, or grape sugar or corn sugar, remember that, and it is known as what? Universal sugar. Okay, glucose is also known as dextrose, grape sugar or corn sugar, and it is also known as universal sugar, because whatever type of sugar you are intaking, complex carbs might be, they will be simplified to glucose and then only the pathway will start, right. So that is why glucose is what? Universal sugar. Next, fructose. Fructose is the most sweetest sugar. It is known as what? Fruit sugar and levulose. So for example, if you are on fruit diet, what happens? Fructose gets isomerized to glucose and then pathway will start. Got the point? Understood? Next. Monosaccharides have free aldehyde and ketone group known as what? Reducing sugars. All the monosaccharides are reducing sugars, accepted, because they have free aldehyde and ketone group. With that free aldehyde and ketone group, they will form N-glycosidic bonds and they turn up forming disaccharides or oligosaccharides or polysaccharides. So when they are forming a bond, they lose a water molecule and they get reduced. That is the reason they are known as what ma? Reducing sugars. So in simple terms, we have learned from our secondary level, okay, that reducing sugars are what? The sugars which has free aldehyde and ketone group. Why are they having free aldehyde and ketone group? To form a bond, N-glycosidic bond, and they get reduced, right. Non-reducing sugars doesn't have a free aldehyde and ketone group. Clear till here, everybody? Next. So you are clear on glucose, fructose and what are monosaccharides. Next, oligosaccharides. So monosaccharides on condensation, if they are turning up forming 2 to 9, like condensation of carbohydrates or monosaccharides of 2 to 9 will form up what? Oligosaccharides. In simple terms, oligosaccharides will have around 2 to 9 monosaccharides. On condensation, they form up what? Oligosaccharides. Examples you see. So what bond is formed between them? Glycosidic bond. It can be 1,4 glycosidic bond, alpha 1,4 glycosidic bond, or beta 1,4 glycosidic bond, or 1,6 glycosidic bond. What does this 1,4 and 1,6 mean? You see here. This is glucose. How you will start numbering from this structure? If you remember, this is C1, C2, C3, C4, C5 and C6. 6 carbons, right? And here also C1, 2, 3, 4, 5 and 6. So first carbon of first glucose is connected with fourth carbon of second glucose, forming up what? Alpha 1,4 glycosidic bond. What is this alpha and beta? If the bond is oriented upper side, that is alpha. If it is to lower side, it is beta. Alpha 1,4 glycosidic bond is formed. Here also you see, this is also alpha 1,4 glycosidic bond between galactose and glucose will form up what? Lactose. Maltose has what monosaccharides ma? 2 glucose. So maltose is a homopolysaccharide, homodisaccharide. Okay, it has 2 glucose unit. Whereas lactose has galactose and glucose. What bond is here? C1, C4. Again alpha 1,4 glycosidic bond oriented towards upper side, right. Here you see C1, C2, 3, 4, 5, 6. So it is also 1,4-glycosidic bond, sucrose. You have to know the composition. Maltose is having what monosaccharides? 2 glucose. Lactose, galactose and glucose. Sucrose, glucose and fructose, sorry. Right? So here disaccharides include sucrose, which has glucose and fructose, maltose, which has 2 glucose units, lactose, which has galactose and glucose, trehalose, which you can see abundantly in insects. This has 2 glucose units. Right? Next is trisaccharides. Example, raffinose, which is made up of glucose, fructose and galactose. Tetrasaccharide, stachyose. Rarely you will get on tri or tetrasaccharides, but it is good or far-sightedness of ours to know about that. Right? So, we have learnt on monosaccharides, glucose, fructose, and we have learnt on what are reducing sugars, and then we have learnt on what are oligosaccharides. Next, examples of that is important, and where you can find 1,4-glycosidic bonds and 1,6-glycosidic bond also important. Right? Next, polysaccharides. See here, you see it will be easy for you. See, there is loss of OH from first glucose and H from second glucose, which is reducing, right, and removal of H2O, forming up of what bond? Glycosidic bond, which is 1,4-glycosidic bond, right. Same the case of polysaccharides, but in polysaccharides, based on the type of monosaccharides that are present, it can be same monosaccharides, it can be different monosaccharides. You have homopolysaccharide and heteropolysaccharide, right. So, these definitions you already know, you know about glycosidic bond. So, we will move on types of polysaccharides, which are very important. So, what is that important you have to learn from carbohydrates? Examples of monosaccharide, examples of disaccharides and what they are composed of, oligosaccharides and then polysaccharides. What are homopolysaccharides? What are heteropolysaccharides? Homopolysaccharides, if you see, they are formed by what? Polymerization of only one type of monosaccharide monomer. For example, starch, chitin, glycogen, cellulose all have only one monomer, that is glucose, right. So, that is what homopolysaccharide. For example, heteropolysaccharides, peptidoglycan, where did you hear this term? Bacterial cell wall, good, very good, bacterial cell wall. It is a heteropolysaccharide. Agar, where did you heard this term? Yeah, second chapter. In plant kingdom, third chapter, where you will algae. Which type of algae will have this type of hydrocolloid? Red algae, right. So, whenever you see any terms which are familiar to you, try to recall it, which enhances your memory towards biology. Okay. So, this is a peptidoglycan and then is agar, right. So, now first we will see on cellulose. Cellulose is a polysaccharide consisting of only one type of monosaccharide, that is what, Nana? Glucose residues only, which are joined by what? Beta 1,4-glycosidic bonds, right. They are joined by beta 1,4-glycosidic bonds. And if you remember the composition of a plant cell wall is composed of what, ma? Cellulose, hemicellulose, pectins and proteins, remembered. So, most of it is composed of cellulose microfibrils, cellulosic fibrils. So, this plant cell walls are made up of cellulose. Okay. Paper made from plant pulp, cotton fiber is what? Cellulosic. This is direct NCERT liner, you have to remember. You need not to mug up this, you just know the meaning. Okay. Cellulose is a homopolymer, has only glucose binded by beta 1,4-glycosidic bond. You can extract it from plant pulp for making paper, and cotton fibrils also have what? Cellulose. Next, cellulose doesn't form any complex with iodine. You can see blue black color of starch test where we have performed in 10th or 8th classes like that. That is forming up, that is responding to iodine and forming a complex, because it has a complex sugar molecules inside it. It has amylose, amylopectin, right? But then cellulose has only glucose units. So it doesn't have complex structures, so it is not forming iodine complex. That's all, right? So these are the points you have to remember with cellulose. Next is starch. Starch is also a homopolymer. It also has glucose, but the bonding is different. Okay, the two units it is composed of is amylose and amylopectin. Amylo has only alpha D glucose, dextro and levo positions that you will learn in chemistry also under biomolecules. So it has what? Alpha D glucose units. It is linked by what? Alpha 1,4 glycosidic bonds. Then amylopectin also it has, that is linked by what? Alpha 1D glycosidic bonds. It also has D glucose units. Okay, so starch is a homopolymer, it has glucose only, but then type of bonding is different. In amylose it has what? Alpha 1,4 glycosidic bonds. Amylopectin it has what? Alpha 1,6 glycosidic bonds. So 80% of starch is composed of amylopectin or amylose. It is composed of amylopectin, and this forms starch iodine complex, giving out what color, Nana? Blue color, right? So you are clear with cellulose, which is a homopolymer of what? Glucose, forming what bonds? Beta 1,4 glycosidic bonds. You can extract it from plant pulp, cotton fibrils you can see, and you cannot you cannot make a complex with iodine. Cellulose cannot form a complex with iodine, whereas starch consists of only glucose. It is a homopolymer, but then bondings are different. You can see amylose and amylopectin. Amylose has what? Alpha 1,4 glycosidic bond. Amylopectin will have what? Alpha 1,6 glycosidic bonds, and this forms a complex, giving out what? Blue color. Next is glycogen, which is a storage food material in animal cells. Starch is a storage food material in plant cells. So this is also a homopolymer, but it forms what? Alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds as well. There is a diagram that is given in your NCERT, wherein the right end is addressed as reducing end, left end is known as non-reducing end. So here you have to observe it like this. This is left end, this is right end. Left and right end, right? Left side and right side. Here can you see free aldehyde and ketone groups? So this is what end? Reducing end. And this side it is non-reducing end. Right? Clear, no? Next, chitin. Chitin is present in the exoskeleton of arthropods. It is the cell wall. It is present as a cell wall in fungi, also known as fungal cellulose. Okay. So this is made up of NAG units, which is N-acetyl glucosamine units, and these are connected by what bonds, Nana? Again 1,4 glycosidic bonds itself, right? So inulin, which is a modified polymer, it is a polymer of fructose. Okay. So what are the sources of inulin? This they will never ask you, but I have given for a general knowledge. Okay. So this inulin will not be absorbed by a human system. Human gut line will not absorb what? Inulin. So inulin clearance is equal to glomerular filtration rate. Where do you see glomerulus? Nephron? Remembered? I remember. Okay. So inulin clearance, it will not be absorbed by the human gut line. So inulin clearance is equal to glomerular filtration rate. That much connection I didn't see, like I didn't expect a question, but then on safe side, that's all. Okay. So biological role, it acts as a storage carbohydrate in some plants. It is not digested by human digestive enzymes. What are the uses in human physiology? This is used to measure what? Glomerular filtration rate in kidneys. It is freely filtrated by glomerulus, not reabsorbed and secreted. So, inulin clearance is equal to what? Glomerular filtration rate. That's all. So, once next is about proteins. Tomorrow we will start. So, once a quickest review on carbohydrates. Carbohydrates are also known as saccharides, whose general formula is CN, H2N, ON, right? And then based on the number of carbon atoms, we have monosaccharides, oligosaccharides, polysaccharides, right? Monosaccharides have just 3 to 5 carbon atoms. They cannot be further reduced, okay, but then they are called as reducing sugars because they have free aldehyde and ketone groups. Like that, tell me a few monosaccharides: glucose, fructose, and fructose, only 2. Maltose is a disaccharide which has 2 glucose units. Galactose, very good. Glucose, fructose, galactose, right? After that, we have learnt on disaccharides: sucrose, lactose, maltose. Alpha 1,4 glycosidic bonds, and what are the monosaccharides it is composed of, just remember. And then we have learnt on polysaccharides: homopolysaccharides and heteropolysaccharides. What are homo and what are heteropolysaccharides? Homopolysaccharides are all that we have learnt: starch, chitin, glycogen, cellulose. Heteropolysaccharides include peptidoglycan and agar. They have a single monomer, okay, but then cellulose has only beta 1,4 glycosidic bonds. Chitin, alpha 1,4 glycosidic bonds, only glucose units are present, that in the form of what units? NAG units, N-acetyl glucosamine units. Glycogen, you can see 1,4, 1,6 glycosidic bonds. Cellulose you have learnt. And coming to starch, you can see 2 branched chains, one amylose, one is amylopectin. 80% of starch is composed of amylopectin, right? And inulin is a polymer of fructose. Good, okay. So, till here we have learnt today elemental, chemical, ash analysis, primary, secondary metabolites and carbohydrates. In the next class we will look into proteins and lipids, right ma? Yeah, thank you.